This video is chapter 3, part 2. It's all about proteins. Proteins are extremely important in the cell because they allow the cell to do a variety of different jobs. For example, proteins allow a cell to get things in and out of the membrane. Proteins allow a cell to move, such as to swim or to engulf food. Proteins allow a cell to divide and replicate, making more DNA and more cells. Proteins also control a variety of different reactions that occur inside of a cell. You will hear about proteins for the entire remainder of the year because they are so important. Every job that a cell does is done by a protein. Proteins are what DNA codes for. So DNA alone is like a set of directions, whereas the proteins are what's built using those directions. Proteins are made of monomers called amino acids. Amino acids are the links that get linked together to make a long chain. The long chain is known as a protein. It's also known as a polypeptide. There are 20 different amino acids that can be linked together in endless ways to create endless amounts of proteins. Similarly, our alphabet, A, B, C, D, has 26 letters, and those 26 letters can be combined in countless ways to produce different words. Same idea. Those amino acids can be bonded together with peptide bonds to create a whole host of different proteins. Different amino acids have different properties. Some of them are nonpolar, meaning that they don't mix with water. These tend to be hydrophobic, meaning they fear water, they don't mix with water. Those amino acids tend to want to be away from water. Other amino acids are polar. Those amino acids we call hydrophilic, meaning they like water and are okay being in a watery environment. Other amino acids are negatively charged, whereas some are positively charged. Amino acids of opposite charges tend to be attracted to one another. The reason that these properties of amino acids matter is because proteins are folded and the shape of a protein is dictated by the properties of the amino acids. Hydrophobic amino acids need to be oriented away from water. Hydrophilic amino acids need to be oriented toward water. And positive and negative amino acids need to be oriented toward one another. Shape is so important for a protein. Shape is king is like a motto of biology. Shape is king. The shape of a protein allows it to do a very specific job. What's shown in this graphic here on the lower right is an enzyme that looks like a blue blob. It needs to bind this particular molecule. So you can see that the shape of the enzyme has to perfectly match the shape of that molecule. If there was a mutation or an error in the shape of that enzyme, in the shape of that protein, where that molecule maybe couldn't bind, that could be detrimental to the cell because whatever products that protein, that enzyme is in charge of making will not be made. Pictured on the left is a protein called ATP synthase. We'll hear more about that in the coming weeks. But ATP synthase acts as a tunnel. So this is like a three-dimensional tunnel through which ions have to move. So if that tunnel gets blocked or if that tunnel isn't wide enough, those ions could not move through there. So shape of a protein is so critical in allowing it to do a particular job. There are 4 levels of protein folding that we're going to talk about that allow a protein to ultimately have a specific shape. So here we go, the 4 levels. Level number 1 is called the primary structure. Primary meaning first. The primary structure of a protein is determined by the amino acids. The code for amino acids is in DNA. So essentially, DNA determines the primary structure. You can see here on the left is a long protein of all these different amino acids. The order in which those amino acids are linked together is determined by the DNA. If there is a mutation or a mistake in the DNA, it is possible that one of those amino acids gets put in incorrectly. Maybe it's deleted altogether. Maybe there's an additional amino acid, or maybe just instead of plugging in a positive amino acid, a negative one got plugged in. Or maybe instead of a hydrophilic amino acid, a hydrophobic amino acid got plugged in. Just one wrong amino acid in the primary structure can cause major issues. One example of this is sickle cell anemia. Sickle cell anemia is a disease that's caused by one mutation in the primary structure of the protein. Whereas normal red blood cells are somewhat donut-shaped, such that they can carry oxygen molecules in the center of that donut, people with sickle cell anemia have misshapen blood cells where there is no little perfect donut shape. So they cannot properly carry oxygen in those sickled, those moon-shaped cells. So this is just one example of how one mistake in the primary structure can cause a major difference in the protein. In this case, it disrupted the protein so much that the entire cell has taken on an odd shape. The second level of protein folding are alpha helices and beta sheets. Alpha helices are a coil. A helix is the same word when we talk about DNA being a double helix. It's a twisted shape. So once those amino acids are sequenced out and linked together, they coil together or they form a zigzag pattern called a beta sheet. That's due to bonds that occur within the amino acid chain, where amino acids start to interact with neighboring amino acids. The third level of protein folding is called the tertiary structure. The tertiary structure is where rules come into play, like those that I mentioned earlier. Hydrophobic portions, hydrophobic amino acids, need to fold in towards the center of the protein. The cell itself is a very watery environment, so by folding the hydrophobic portions toward the inside, those amino acids are kept away from water. Hydrophilic portions of the protein, those that do interact with water and are okay with water, will end up pointing outwards. And then, like I said previously, positively charged amino acids pair with negatively charged. This is why one wrong mutation, one wrong amino acid can greatly change the shape of the protein. The fourth level of protein folding is where one entire chain interacts with another entire chain. This particular picture shows that there are multiple chains all interacting. We call that the quaternary or the fourth level of protein folding. To recap, these 4 levels are so important because they determine the shape of the protein. The shape is critical. Primary structure, sequence of amino acids. Secondary, alpha helix and beta sheet. Tertiary is where we follow those pairing rules. And then D, quaternary, we have an entire chains that come together. Here's some more examples of why protein shape is important. When we talked about bacteria and quorum sensing, we talked about how bacteria cells can communicate with one another. They do so by releasing little tiny molecules into the environment. A protein, represented by this blue box, is responsible for binding that signal. If there is a mutation in that protein such that that little molecule cannot bind, the cell would have no way to communicate with neighbors. Similarly, that's shown in all three of these pictures. Protein shape is important. Improper folding of proteins can cause a whole host of diseases. Listed here are just two examples, but cataracts are where eyes start to look cloudy. It's due to an accumulation of misfolded proteins. Generally, people who get cataracts are elderly because they've lived their whole life with their DNA, and then things start to deteriorate and proteins can no longer fold properly. Similarly, in Alzheimer's, there are neurons that need to fire and communicate throughout your body. Well, in a patient who has Alzheimer's, there's chunks of proteins between those neurons that block them from communicating. So these are just two examples of why protein folding is important. When a protein unfolds, we call that denaturing. This is a word you'll see often. Denaturing is when a protein unfolds. There's a few things that can cause that. One of them is heat. Heat is energy. If I have a nice folded protein and it is heated to a high temperature, it causes the protein to start to move more. As the protein is moving more, it disrupts bonds that are occurring within the protein such that those bonds between amino acids break and the protein essentially unfolds. Another example that can cause denaturing is pH. If I put a protein in a very acidic solution, an acidic solution has a lot of hydrogen ions, meaning that protein becomes surrounded by positive charges. If there was a little amino acid who was very negative and it was paired with an amino acid that was very positive, that negative amino acid might no longer be attracted to the positive amino acid, but rather it's attracted to a positive in the environment. That then would break the bond between the positive and negative amino acids because the negative amino acid is becoming attracted instead to the positives in the solution. The opposite is also true for a basic solution. We also call basic alkaline solutions. Alkaline solutions have a lot of hydroxide ions. So the protein, if it's put in a very high pH, a very basic pH, I would have all these negative charges in the environment, and an amino acid that used to be attracted to a negative amino acid might now instead be attracted to the negatives in the solution. Salts work in a similar fashion. Salts are made of positive and negative ions. So the idea behind salt is the same as that of pH. The amino acids become attracted to the charges in the solution instead of to the amino acids within the protein.